the invitation. Excuse me. Thank you for the invitation to share what we're doing here at the Beckman Foundation. Just get the slides up. Hopefully that's visible to everybody. I think we'll be a little bit different than the government presentation that we just heard. This is a work at a private foundation founded by Arnold and Mabel Beckman Foundation, and hopefully the Beckman name is fairly familiar to this audience in terms of, Arnold Beckman started a company back in 1930s where he made one of the first electronic readout pH meters, and that company really took off, and he was a very successful businessman and created a international large company for Beckman Instruments that later became Beckman Coulter, supplying instrumentation to laboratories around the world. And so what we do at the foundation that they established is we really try to continue on that legacy. So Arnold and Mabel Beckman in 1977 made a decision to leave their personal wealth in a foundation that would last in perpetuity. And the mission, as you can see here, is to support science. So, Dr. Beckman always said, he was very lucky and made a lot of money and was very successful based on science. And he wanted to be able to give back into that scientific community that supported him and his family through the years. And in addition, there's a quote there at the top: "I have done more for science by making instruments available than I could have done working in my own laboratory by myself." And this is really part of the core of what we do here at the foundation and carrying forward his legacy, is to say, okay, when you bring instruments in, you enable many different types of experiments to be done. So there are, we do also run grant programs which are very specifically focused on a particular research project area. But what I'm going to talk about today are instrument grant programs, where we're really carrying forward this part of the Beckman legacy, where by enabling instruments to be brought into laboratory spaces and not constraining them necessarily to a particular project, we can really help push forward large areas of innovation and discovery. One of the other things, especially with the Beckman Foundation, that we're very interested in is young investigators. For all of our grants, you'll see that there's a requirement that very junior people are included. There's lots of training that's included. This is something that Dr. Beckman very much wanted within any grant portfolio that we run. One of his sayings also is that he wanted people who didn't yet have the clout to get major research grants from the federal government. Those are the people he wanted to support. And so we're always looking to bring that element into our grant programs as well. So we have run a couple programs in the recent past I just wanted to highlight here that are really geared towards this instrumentation type of grant. So back in 2017, we had a fairly large program, about $12.5 million total investment from the foundation. And with that, we gave 5 awards for cryo-EM. Those were to enable groups to purchase a Titan Krios or other similar types of instrumentation. Obviously, those are very expensive instruments. You might say 5 divided by 12.5 is actually only 2.5. So they were $2.5 million awards a piece. And so there was a lot of cost sharing involved with the organizations that received these awards. And it was to purchase, install, and then do 5 years of maintenance, a commitment for 5 years of maintenance for high resolution cryo-EM. We then, in 2021, ran a program on light sheet microscopy. And for this, we gave out 8 awards. And really the key for this program was not only to look for those microscopes and the teams that could really bring in this new technology, but to also say, we really want you to bring in a data scientist as a core team member so that, with these large, long-term 3D movies that you're acquiring now with this new technology, how are you going to do all of the data analysis that's required to actually get to discovery rather than just the operation and usage of the microscopes? And then this past year, and actually today, I think is the kickoff of the awards that we just announced for our last program here on the list, which was to bring in sample prep for cellular cryo-ET, and specifically looking at FID milling. And really in this program, we were looking to see, you know, how can we help get more people into the field using this kind of technology so that maybe the effectiveness of the technology could be improved. How can we get innovation into the sample prep workflow that is bringing really stunning images out, but is very sort of low throughput, high intensity right now. So how could we help in ways to bring innovation into those workflows? And so that was the focus of the grants that we are just getting started. So, you know, internally, I just wanted to share a little bit of sort of how do we do this for this group. So it's not a mystery. So, you know, we go through a process, needs to mute. Internally, we have a program development process. So we have a group of a science advisory council, which is 7 individuals that, you know, advise the board of directors and the foundation staff. These are people who are professors at universities. Many of them won some of our individual grant awards in the past, and, you know, donate their time and effort to the foundation to come up with broad themes. So what we're looking for are, you know, where are great innovations happening right now, where instrumentation access is sort of a bottleneck. And so we have this Science Advisory Council group come up with these broad themes. And then what we've done in the past couple of years is we pull together some workshops. Based on those themes, we bring in some people who are experts in those areas and kind of hash out, well, you know, what would a Beckman investment in this look like? What might be the impacts? And then we generally end up with a laundry list. And from there, we start to say, okay, where are the places where we're seeing gaps in funding from other agencies potentially, where Beckman could really make a big impact. So we've had a couple of these workshops. I actually anticipate that we'll be starting the planning process for the next one, you know, during this year to potentially host next year. Maybe we can get back in person. We have continued this with the Zoom capabilities as everyone else has. And so this isn't to say that I have anything to say, hey, be on the lookout for this yet, because we really are sort of in that initial planning process. So I do anticipate we'll have some future programs coming and those announcements. So when we do have a program out, just put up here our website and our application portal. You can check those places. We do make a active outreach into communities of interest for particular project areas as best we can, getting the word out there that we've got an opportunity open. And we try to keep our applications very simple. So it's going to be a couple pages. So you got to keep it simple and keep it short in terms of your proposal. And I would say, one thing, and you have to kind of know who you're applying to when you apply to a private foundation, but the training and the incorporation of junior scientists is really just essential to us. And so we do ask you to take some time and tell us how that's going to happen. And so please, if you are putting an application into us, make sure you do pay attention to that section. That's something that's very important to us. And we always list, people that you could reach out to. And we really are, we're here to help. So please reach out. We'd love to get quality applications into these programs. So don't feel bad about asking some questions. And then just to say, if you would like to hear about these opportunities that we have, please go ahead and send an email. That's instrumentgrants@beckmanfoundation.org. I do monitor that email. So if you have any questions or follow-up or interest in future programs, please feel free to drop a note into that email and we'll save your contact. The one thing that is just essential to us is to understand who's in your research support office as well, the people who are going to help you give those grants. So if you don't mind also including that information, if you could reach out, that would be very beneficial to us. I think that was it. So I'll pass it back to James.
Thanks, Anne. That was wonderful. Thank you so much. Vlad, I'll pass it over to you.
Thank you very much, James. Thank you for the introduction, and thank you for the invitation and giving us an opportunity to talk about what we do at CZI. It's great to be here. I see a lot of familiar names, and I hope to establish new connections as a result of this talk. Can you see my screen?
Yes,
Terrific.
we can see your screen. Thank you.
Terrific. Thank you. So let me start with a brief overview of CZI. So Chan Zuckerberg Initiative was founded in 2015 by Dr. Priscilla Chan and Mark Zuckerberg to help solve some of society's toughest challenges, from eradicating disease and improving education to addressing the needs of our local communities. The mission of the Chan Zuckerberg Science Initiative is to support the science and technology that will make it possible to cure, prevent, or manage all disease by the end of the century. While this may seem as an audacious goal, over the last 100 years, biomedical science has continued to make tremendous strides in advancing human health and treating disease. A comprehensive understanding of disease onset and progression requires quantitative assessment of molecular events across a wide range of spatiotemporal scales and organizational complexities. Imaging uniquely offers this capability. To that end, to achieve this, we're working on 4 directions. In Frontiers, we directly support development of disruptive technologies to view biology in context. To further accelerate innovation, last year, CZI has announced that we will now establish the Chan Zuckerberg Institute for Advanced Biomedical Imaging. It's going to be a physical space where multidisciplinary teams will come together to develop revolutionary new imaging platforms. Now, to fully benefit from this technology, we'll also need well-trained experts, points of dissemination, such as shared imaging facilities. We will need a collaborative community to develop standards, to create platforms, to share protocols, tools, and methods. This is the core of the community initiative. Here, we support imaging scientists all over the world to manage imaging core facilities, come up with innovative educational approaches, develop standards, and work closely with biomedical researchers to help to advance research with the new technology. Finally, to complete this workflow, in Analysis Thrust, we work to provide universal access to reproducible quantitative imaging analysis. A little bit about what we have achieved in each of these thrusts. In Frontiers, as many of you are aware, there's always a problem of choice and priority. On one side of the spectrum of resolution, we have methods that allow us to image at high resolution, but only up to 1 millimeter deep in tissue. Super resolution, confocal microscopy, lots of examples. On another side, we have imaging methods like MRI, X-ray, and ultrasound that allow us to go deeper, but at the same time, the resolution suffers there, and it's well lower than the cellular resolution. The goal of the deep tissue imaging program that was announced about 2 years ago was to support the technologies that would enable us to image through tissue and bone, and yet maintain and preserve the cellular or ideally subcellular resolution. There were 13 pilot projects selected with a total budget of 13 million. These projects, they span across 7 imaging modalities, going all the way through MRI, X-ray, light microscopy, photoacoustics, and extend to computational methods, such as, for example, quantum imaging. The funding is provided for initial 2.5 years as a pilot projects. And once they're completed, the successful pilot projects will be reviewed and they'll progress to phase 2. During phase 2, the teams will be able to rotate in order to enable a truly multi-expertise teams. And then the next goal would be scaling and dissemination of this instrumentation. On another side of the resolution spectrum, we have the program Visual Proteomics. Here, the goal is to support the technologies that would enable visualizing proteins inside cells, how they form, how they change the structure, interact with each other. Currently, this is the primary domain of electron microscopy, but visualizing proteins in cells is still challenging. So the goal with visual proteomics was to support the technologies that would enable to do that. We have selected 14 projects. Here, the types of the projects, they span across correlative microscopy, hardware, software, and probes development. Given that the cost of electron microscopes is usually quite high, it's cost prohibitive, and dissemination, instead of mass dissemination, during phase 2, we'll focus on creating the central hubs that would enable access to these new technologies to a broad community of biomedical scientists. Now, a critical role in the technology's validation and dissemination belongs to imaging scientists, imaging facility operators, managers and directors, image analysts, imaging software developers, and others with a unique expertise necessary to advance biomedical sciences with imaging. Despite the essential role of the science community, imaging scientists now have very few ways to secure funding. This was the very first grant of the imaging program aimed at supporting salaries of the imaging scientists. The length is 5 years with initial 3 years and potential extension with another 2 years after the review. We already had the second cycle with a couple of years ago. In total, we had a budget close to 35 million in support of 40 key imaging experts, supporting their local communities of biomedical researchers and supporting global imaging community. The philosophy here is to close the gap and attract other funders. Importantly, each had an agreement where the host institution will have to commit to continue funding of the imaging scientists when the program is over. The program was open to imaging scientists pretty much globally with just very few exceptions. You can see them on a map marked with red dots. You can see that we have quite a lot of grantees in North America. They go all the way to Europe. We have grantees in Australia. There are a couple of grantees in Latin America and Africa. Accelerating science is a team effort. It requires coordinated collaboration. Network organizations, they bring together imaging scientists for discussions, brainstorming, coordination to avoid duplications and amplify the effort. We are very happy to support Bioimaging North America for the fantastic work the organization is doing on so many fronts in imaging education, creating information systems for imaging scientists, work on standards and more. There are other organizations in the world with the goal to foster regional imaging communities, and the coordination between all of these organizations is done by the umbrella organization, the Global Bioimaging. They are working on an agenda that would bring together imaging scientists all over the world to discuss their challenges, and sometimes specific to the region, and creates a platform for collaboration, global support, exchange of experience. Now, in our previous programs, in particular the imaging scientist program, as you can see from the map, the distribution of the red dots, we didn't get many applications coming from regions like Latin America, Africa, former Soviet countries. And we reached out to multiple imaging scientists, biomedical scientists, key opinion leaders in this region, conducted over 50 interviews. And based on that, we built another program that was better tailored to the needs of the imaging community in these regions in order to advance bioimaging. The result was the Expanding Global Access to Bioimaging program. Last year, we have selected 17 grantees with the projects that are aimed at advancing bioimaging in 31 countries. You can see them marked in yellow, and are focused at building capacity among biomedical researchers, fostering regional community of imaging scientists to coordinate shared usage of the existing resources, and also training the new cohort of imaging scientists that would be able to help biomedical researchers with the access and utilizing the full capabilities of advanced imaging methods. With both of our grantee groups in Frontiers and Imaging Scientists, we consistently hear that image analysis is one of the biggest bottlenecks for them. We also support software developers for open source packages like Fiji, Scikit Imaging and Cell Profiler, and have partnered with the Open Science program at CZI to support software developers of essential open source packages. There are a total 19 projects supported within that program. Our imaging SciTech team Our imaging SciTech team is building out an image analysis platform and incentivize community developed plugins to help bring quantitative image analysis to the broader ecosystem. Here you can see Napari. It's a community-built Python-based open source tool designed for browsing, annotating, and analyzing large multidimensional images. The Napari Plugin Accelerator grantees, they were selected last year. They will join a community of researchers who are contributing to an extensive plugin ecosystem to provide easy access to reproducible and quantitative bioimaging analysis. These grants will support development and maintenance of Napari's growing ecosystem of plugins. And Napari is not barely just a viewer, it's a platform that allows a very easy integration or writing an algorithms, Python-based algorithms, to embed that to the existing graphical user interface. To continue building on that, our team has enabled the Napari Hub. This is the place that brings together the algorithm developers and biomedical researchers who want to use them. Here, biomedical researchers can easily browse through the plugins sorted by the application, by the technology utilized, by the category, and integrate that into Napari for their own workflows. One other aspect of our support is clinical imaging. We have one initiative in the clinical space funding the UK Biobank. It's a repeat imaging project. This is an unprecedented opportunity to contribute to a large-scale discovery data set for human medical imaging. It encompasses structural and functional brain imaging, whole body MRI imaging, cardiac MRI and ECG, Doppler ultrasounds for vascular structure pathology, and DEXA imaging of bones, joints, of hips, the spine. There are 70,000 volunteer participants. They will be imaged at 2 different time points, 2 to 7 years apart. Their imaging data will be integrated with electronic health records, genetic data, and other information such as survey responses. This is not just an unprecedented medical data set of healthy volunteers. It is also an excellent opportunity to follow disease progression, starting from the healthy state. For instance, based on the models of disease incidence, it is expected that 6,000 cases of dementia or Alzheimer's and 1,000 cases of Parkinson's disease will be diagnosed in this 70,000 person cohort during the period of the study. Based on this data, we'll be able to develop the phenotypes of the disease onset and a progression that can be used in the future for informing very quick diagnostics and treatment. A quick overview of our team. The imaging program is led by Stephanie Otte, who sets the strategy and supervises the entire team. My colleague Stephen Jett, he manages or leads on a frontiers end. Myself, I'm focused on imaging community development. Melissa Gorham is our project manager who enables all of the knowledge management and make sure that we're on track with all of our projects. Our SciTech team engages with the image analysis community and works together with them to develop Napari. Nick Sofronio leads these efforts, and Justin Kiggins is the product manager for the Napari Hub. Now, this is the final slide where you can see all of the contacts that you can use in order to get in touch with us. We're trying to stay very close and with the community. I would love to hear your feedback about the needs of the community, or have discussions about your ideas, how the community can evolve, how can we advance the bioimaging with the community. So feel free to reach out. If you want to stay aware of all the funding programs that we have that are coming, please sign up for our CCI newsletter, and you can read more about the projects that we have run, the overview that I just gave, on our webpage with the link here showing. Thank you. Happy to take any questions.